Thank you so much for the introduction, and thanks everyone who decided to join us today to celebrate the achievements of the Biohub. I'm going to be talking to you about our work on building all organism cell atlas. If we could go on the next slide, please. So the Biohub vision, which is very much in line with the CZI mission, is to cure all the diseases by the end of the century. But before we can actually start to intervene, we first need to understand what does it mean to be healthy, such that we can infer what goes wrong upon a perturbation, and then we can actually intervene to treat or manage that condition. So with that in mind, we decided that we would start by building reference atlas. And our first attempt, Tabula Muris, actually maps the cells of the mouse, because the mouse is the most commonly used model organism in biomedical research. But as I mentioned, we want to be able to use this reference atlas. So when we are putting our toolkit of computational tools that will allow us to query the atlas, we need to be able to understand what changes given a perturbation. So we took advantage of natural aging in the mouse, and we built Tabula Muricinis, which is a compendium of cell types of the mouse across the lifespan of the animal that characterizes over 350,000 cells of the major 23 organs and tissues of the animal. And while we were doing this atlas, we really enable the use of single-cell transcriptomics in our surrounding community, and have been part of major consortiums that are now mapping other organisms to really build a comprehensive catalog of reference cell types across different species and diseases. And for today's talk, I've decided to focus on our most recent effort, which is Tabula Sapiens. If we could go on the next slide, please. So I want to just stop here for a bit and call your attention to one of the strengths of the Tabula dataset, which is we pay a lot of attention on how to minimize the technical artifacts during the sample collection. And so when we were doing the mouse datasets, there was order by which the tissues would be harvested from the animals to ensure the best quality possible of the data. With the human, we tried a very similar approach in the sense that we've partnered with Donor Network West, which is a nonprofit organization that facilitates the procurement of the organs and tissues for research. And we try to procure as many tissues as possible from a single individual, and then speed up the time that it takes from the tissue procurement until we start processing the data. If we can go on the next slide, please. So this really looks like two major blocks. So in one hand, we have the procurement of the tissues, and then the organs get shipped directly from the operation room into each of the respective expert labs, where they get processed using the best state-of-the-art protocols that the experts have developed to get from the full organ into a cell suspension. But then all this data is centralized at the Biohub, and then the rest of the experimental procedure and the data analysis is done in a consistent manner, which ensures a highest quality control over the data, higher consistency, and hopefully yields better quality at the end. So if we can go on the next slide. Here I'm showing you just the bird's eye view of where we are as of now with Tabula Sapiens. This is a growing data set. So far, we have annotated over 480,000 cells across 24 organs, and these come from 15 donors. From 4 donors, we were actually able to procure at least 5 organs, which allows us to really start asking questions about the diversity within an individual and across the population. And then side by side with our single cell transcriptomic profiles, we are also sharing clinical metadata, which is going to empower us and the community to start correlating patient characteristics with the single cell transcriptomic profiles. Can we go on the next slide, please? So the previous slide kind of like gives you the overall summary of the data, but how do we get there? It has taken us a couple of years to put this in place, but what you are seeing here that I'm not going to go into much detail, but I'm happy to discuss offline, is by now a fairly standard workflow from which we go from raw counts after the sequencer into a joint embedding space where we can start to visualize how does the data look like. Next, please. So at the end of this initial pre-processing step, this is fairly automated, where we remove the contamination of the RNA, we correct the gene count, and then we kind of like embed all the data together in a 2D visualization. We can color by variables that we are certain of, such as the organ of origin, or if we go on the next slide, for example, the donor, can we go next, please? The donors from where each organs and tissues are coming from, or any other metadata we might have available. But actually, for us to be able to use this atlas, we need to take a step further and annotate each of these individual dots that we're looking here in all these different colors according to the metadata, which actually correspond to an individual cell. So on the next slide, please. The next part of the workflow is the annotation of the data set. And because Tabula sapiens is a growing data set, we have decided to take a mixed approach. So the first time we bring a tissue into the atlas, we ask the tissue experts to carefully annotate the cell types for that tissue. And then we process all the data together using an algorithm that I'm going to go a bit more in detail at the end, which is popular vote, or POPV in short, with the computationally to annotate all the data set. And then we ask the experts to review. And so we iterate on this loop as many times as needed for us to build confidence in the data set. And so far we have annotated for 175 cell types, but what's really unique about the Tabula data sets is all of these have been curated by the tissue experts, which really gives us high confidence on the data. If we can go on the next slide, please. So we have really leveraged the power of single-cell transcriptomics. Is a scalable, powerful technology, but we are at the point with Tabula sapiens that we feel we could really grow the modalities that we make available with the atlas. And so part of the Tabula sapiens consortium, we now have a pathology team that is helping us to understand how does the tissue actually look and how good is our recovery of the cell composition with single cell. If we can go on the next slide, please. So the pathology team has carefully annotated some of these example images. So here on the left, I'm showing you the intestine H&E stainings, and you can see all the cell types that the team was able to annotate. But this is very hard to do just from an H&E staining. So working together with them, using an Apari plugin, we asked the team to, instead of annotating all the cells, to actually quantify the abundances of the functional compartments. We can go on the next slide, please. So we have estimates of what do we expect to obtain in terms of the amounts of endothelial cells, epithelial cells, and so on. So now that we have this picture of this is what we know the tissue should look like, because this has been quantified from undissociated tissue, we can ask how good of a job are we doing when we just look at the dissociations of the cell types. If you can go on the next slide. So when we correlate the cell types that we obtain from the single cell sequencing and what we expect from the pathology, we actually find a good correlation between the two modalities, which boosts our confidence that our data set and our atlas is really comprehensive and a reliable picture of the organs that we have. If you can go on the next slide, please. And another modality that we thought would be complementary and really kind of like coming around and rounding up the atlas or for Tabula sapiens is the microbiome. If we can go on the next slide, please. I'm going to go very briefly in here. But so we looked at the diversity of the species across 5 regions of the intestine. And what we found, if we can go on the next slide, please, is that very much in line with what we also mapped on the cell types using the single cell transcriptomics. There is a much higher diversity of microbiome species and cell types in the small intestine, while on the large intestine, so between the column, the diversity of the microbiome species and the cell types is much smaller. And I'm happy to discuss this further. If we could go on the next slide. Now I'm just going to transition here to give you a feeling of some of the tools that we've had to build to really be able to create these large reference data sets. And most of our time has actually been investing on annotating the cell types, because we know that this is a very time consuming task, but if we don't do it well, all the analysis, because they depend on the cell type assignments, they're not going to be correct. So it's really important we get this right. And so we've collaborated with Biohub investigators and within the Biohub teams to create original methods that help us using previously annotated data sets to annotate new ones. But what we are now ready to do, because we have these comprehensive references with Tabula muris, Tabula microcebus, and Tabula sapiens, if we can go on the next slide, please, is really to put out this reference as the database against which anybody can annotate their data sets. And so we have an interface that people can log in and they can upload their data and get an automatically annotated data set depending on which species they are using. And this is a collection of tools. So this is POP3, the same algorithm we're using to annotate Tabula sapiens. We look at this as a bundle that we can easily add or remove methods. And so it's going to be scalable and we can keep up with the new developments in the community. I can go on the next slide, please. The next very important aspect of these data sets is to how do we explore them? How do we really use it? And for that, we've been very fortunate to work closely with the cell by gene team from CZI, who have built a portal to which it's very easy for anyone in the community to go to and start interacting with the data. We make our data sets available through the cell by gene data portal. But we also use cell by gene not just as this exploration tool. This is more when we share the data with the community, but really as our working tool. So if we can go on the next slide. When we get the data, if you remember a couple of slides ago, when I said we have, okay, this fairly standard by now workflow until which we can get a map of a 2D embedding of the cells, this is how it looks like. It doesn't have any biological insights, but using cell by gene, somebody that is not computationally savvy necessarily can quickly start looking for known genes or interesting findings. So if we go on the next slide, in this example that I'm using today for this quick tour, I'm showing you the tongue. So somebody can look for the keratin genes, for example, and they can look for a single gene, for a set of genes. And if they find interesting cluster of cells here, they can put on their annotations, as we can see on the next slide, that any type of metadata annotation, including the cell types, can be easily done using the cell by gene tool. And this is really accelerating how we build the data sets by a factor of 10 to 20 times. So something that used to take months is now we can do it in less than a week. In talking about accelerating science, if we can go on the next slide, this is something that is core to the Biohub. We really want to accelerate scientific breakthroughs. And the way we are changing our data, data sets are shared within the community is by making sure all our data is easily accessible using data portals. All the versions of our manuscripts are posted on bioRxiv. We release the code we use for the analysis together with the raw data and processed data. And on the left, you have kind of like the history of how we did it for Tabula muris, but I want to call your attention to the right. So for Tabula sapiens, we took a step further. And before we had the preprint, we released the data for the entire community to use. So people could start doing their own analysis even while we were still preparing the preprint. And we have done the same for Tableau Microcebus. If we can go on the next slide, please. So I want to wrap here and thank all our partners and collaborators. Thank you all also for listening. The Tableau projects are true examples of team science. These consortiums here are over 150 people, and we have quite a lot of groups that are part of multiple consortiums, really speaking to the value of the data sets, because these grad students and postdocs and us at the Biohub really put a lot of effort in making these references that are useful for the community to use. And I hope you go and explore our data set. If we can go on the next slide, please. These are just a set of links. They are also available from the Biohub website. And I'm happy to take any questions.
Angela, thank you very much for a fabulous talk. We have a question from Sarah Suleiman, which is for Tabula sapiens. Is the cohort genotyped to allow for instance, looking at genetic effects that might affect gene expression from individual to individual?
No, we do not have DNA sequencing of the patients. And we ask the users of the data set to sign ethics agreement in terms of like the usage of the data.
Okay, great, thanks. And then can you describe a little bit the tools that are currently available and also new tools that you may be thinking about, both to allow data visualization to find patterns within the data and new ways of visualizing the data that can enable drawing connections with other data sets or generating to facilitate generating new hypotheses based on these data?
Sure. I said that we have been really leveraging the cell by gene as a tool to which we can quickly draw some insights into the data. And we are working on methods that allow us to put data sets together that we can interrogate some of the shared patterns across the different data sets. In terms of tools itself, we are mostly consumers so far of visualization tools like heat maps or networking maps, but that is something that we are interested in building further, is really a way of asking these questions of like, when we see a certain cell type in one of the organs, how can we quickly assess if this is common across different donors, or if this is kind of like a donor specific. So these are the type of questions that we're trying to solve now that haven't been completely addressed.
Great, Angela, thank you very much. And thank you again for a great talk.